Reactions of lactose. First, we will be performing solubility test. Test the solubility of the given substance in water. It is turned out to be soluble. That indicate absence of polysaccharides. Then Molish test we will be performing. Take 1 ml of test solution in a clean dye test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 drops of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of the two liquids. Presence of carbohydrate. It is dehydrated by concentrated H2SO4 to form hydroxymethyl furfural or furfural, which condenses with alpha naphthol to form a violet ring at their junction of the two liquids. Carbohydrates, concentrated H2SO4. You could see this reaction. Then Fillings test. Fillings test. To 1 ml of test solution, add 1 ml of each fillings A and B solutions and mix it well and heat in a boiling water bath for 3 minutes. Reddish brown colored precipitate is obtained. Presence of reducing sugar in alkaline medium. The carbohydrate with free aldehyde or ketone group reduces the cupric ion, cuprous ion. The cupric hydroxide formed is converted to the cuprous oxide which gives red precipitate on heating. Then Benedict's test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution and boil it for exactly 2 minutes. Brick red precipitate. Presence of reducing sugars in alkaline medium. The carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. The COH formed is converted to the cuprous oxide form which gives red precipitate on heat. Then nitric acid test. To 1 ml of the test solution, add 1 ml of concentrated HNO3 and heat in a boiling water bath till the brown fumes stop evolving. First, 1 ml of the test solution, add 1 ml of concentrated nitric acid and heat in a boiling water bath till brown fumes stop evolving. Keep it overnight and observe the crystal under microscope. Broken glass piece-like crystals are obtained. Presence of lactose. Then Osazone test. Take 3 ml of test solution in test tube and add 1 spatula of phenylhydrazine hydrochloride. Add equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in a boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Then take out the crystals with the help of a glass rod. Mount on glass slide using cover slip and observe under microscope. You could see powder puff or hedgehog shaped crystals. In case of lactose. In presence of lactose, glucosazone, lactose react with phenylhydrazine, sodium acetate mixture to form lactosazone. Then next we are going to learn about reactions of maltose. Solubility test. Test the solubility of given substance in water. Soluble, absence of polysaccharide. Then Molish test. Take 1 ml of test solution in a dry clean test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of two liquids. Presence of carbohydrate. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condenses with alpha naphthol to give violet ring at the junction of the two liquids. Then Fillings test. To 1 ml of test solution, add 1 ml of filling solution A and B. Mix well. Heat in boiling water for 3 minutes. Then reddish brown precipitate is formed. Presence of reducing sugar in alkaline medium. The carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. The COH formed is then converted to cuprous oxide and which give red precipitate on heating. Then Osazone test. Take 3 ml of test solution in a clean dry test tube and add a spatula of phenylhydrazine hydrochloride and equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in a boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Then take out crystals with the help of a glass rod. Mount on a glass rod to a slide using cover slip and observe under microscope. We could see sunflower shaped crystals, presence of maltose. Earlier we mentioned a trick to remember this: maltose, M, marigold. Marigold is a flower, sunflower is another flower. So MS, like that. Maltose, sunflower shaped crystals. Then we will be performing oxygen test. After we got sunflower shaped crystals that indicate presence of maltose. Maltose reacts with phenyl hydrazine sodium acetate mixture to form maltosazone, which confirms the presence of maltose in test tube solution provided. After that, we are going to learn about reactions of polysaccharide. So far we learned some major reactions to test the presence of monosaccharides and disaccharides. Then later we are going to learn about reactions of polysaccharides. So polysaccharides are the macromolecular polymers of monosaccharides units linked by glycosidic linkages. On the basis of the type of monosaccharide unit present, the polysaccharides are further classified into homopolysaccharides, starch, glycogen, cellulose, chitin, etc., and heteropolysaccharides, heparin, hyaluronic acid, keratin sulfate, starch, etc. Homopolysaccharides, in the sense their monomers will be similar or same, but heteropolysaccharides, their monomers will be different. For example, heparin, hyaluronic acid, keratin sulfate, starch, etc. In plants, particularly in tubers, roots and seeds, are these polysaccharides present as reserve food stuffs. Then it is made up of amylose and amylopectin. Amylose is a straight chain polymer of glucose molecule which form helical structure and it is water soluble. Amylose and amylopectin. Amylose is a straight chain polysaccharide polymer of glucose molecule which form helical structures and is water insoluble. And Molish test. We are going to perform Molish test. This is a general test for all carbohydrates we mentioned earlier. So carbohydrates, when treated with concentrated H2SO4, undergo dehydration to give furfural derivatives. Hexoses, for example, forms hydroxymethyl furfural. These compounds condenses with alpha naphthol to form colored products. Under acidic conditions, the polysaccharides are hydrolyzed to monosaccharides which answer the test. Then later we will be performing Benedict test. Benedict test in the sense reducing sugars in alkaline medium tautomerize and form indiols. The 1,2 indiols will give formaldehyde and pentose. The chain reaction continues to produce short chain aldehydes which are powerful reducing agents. They can reduce cupric ions to cuprous ion, which is a basis for Benedict's reaction. The cupric hydroxide formed is not easily soluble. In order to keep the hydroxide in solution, a metal chelate like a citrate or a tartarate is included in the solution. So Benedict solution contains the milder alkali Na2CO3. When reducing sugar solution is heated with Benedict's reagent, the sugar is decomposed and end products introduce the cupric ion to cuprous ion, a yellow cuprous hydroxide. Cuprous hydroxide produced during the process of heating is converted to red cuprous oxide, 2CuOH, Cu2O plus H2O. Then iodine reaction. Remember, we are performing tests for polysaccharides. So some new tests are involved in this scheme, like iodine reaction. The amylose component of starch has helical structure. When it is treated with iodine solution, iodine is trapped inside of the coil and complex has an intense blue color. When starch solution is heated, the helical conformation is disrupted and it loses capacity to bind iodine on cooling. The original conformation is required and the capacity to bind is also reverted. Then amylopectin and glycogen, because of branched structure, gives light purple or red color with iodine. Then Benedict tests an acid hydrolysis. Starch and dextrins are hydrolyzed by hot dilute mineral acids to yield glucose, which answer Benedict's reaction. To 1 ml of starch solution, add 5 drops of concentrated HCl. Boil the solution for 2 minutes. Cool it and neutralize hydrolysate with the 5 drops of 40% NaOH. Hydrolysis reaction is performed. Then check using a piece of red litmus paper. Perform Benedict's test with the neutralized solution. The starch and dextrin gives positive test. And reactions of starch in particular. Then first we will be performing solubility test. Test the solubility of a given substance in water, partially soluble. That indicate presence of polysaccharides. Then Molish test is performed. Take 1 ml of test solution in a clean and dry test tube and add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 drops of concentrated H2SO4 along the sides of the test tube without shaking. You could see the creation of a reddish violet ring at the junction of two liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural derivatives which condense with alpha naphthol to form H2SO4 to form furfural derivatives which condense with alpha naphthol to form a violet colored ring at the junction of two liquids. Try to write this reaction. An iodine test is performed. Take 2 ml of test solution and add 2 to 3 drops of iodine solution. Heat and then cool under tap water. Deep blue color disappear on heating and reappear on cooling. Presence of starch. Amylose component of starch has helical structure. When it is treated with iodine solution, it is trapped in coil and gives intense blue color. On heating, helical conformation of this is disrupted and it loses its capacity to bind with iodine. On cooling, the original conformation is recovered. Okay. Then Benedict test. Take 5 ml of Benedict's reagent in a test tube and add 8 drops of test solution. Mix well and boil it for 2 minutes. No colored precipitation. Starch is not a reducing sugar. After that, we will be performing acid hydrolysis. Take 5 ml of test solution in a clean dry test tube and add 4 drops of concentrated H2SO4 and keep it in a boiling water bath for nearly 10 minutes. Cool under running water and then neutralize by adding 5 drops of 10% sodium carbonate till the effervescence ceases. Okay. The solution is hydrolyzed. Later we will be repeating this Benedict's test after hydrolysis, acid hydrolysis. Take 5 ml of the Benedict's reagent in a test tube, add 8 drops of hydrolysate and test the solution after hydrolysis. Mix it well and boil it for 2 minutes. That create brick red precipitate. Hydrolysate contain reducing sugars. Then reactions of dextrin. Solubility test. Test the solubility of a given substance in water. Partially soluble. Presence of polysaccharide. Molish test will be performing. Take 1 ml of test solution in a clean dry test tube. Add 2 to 3 drops of Molish reagent. Mix it well and add 1 to 2 drops of concentrated H2SO4 along the sides of the test tube without shaking. A reddish violet ring at the junction of two liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural derivatives which condenses with alpha naphthol to form a violet ring at the junction of the two liquids. Then iodine test. Take 2 ml of test solution and 2 to 3 drops of iodine solution, violet black color. Dextrin is a polysaccharide. Amyloerythro or archodextrins formed of intermediates during hydrolysis of starch gives violet, red and no color with iodine respectively. Unknown carbohydrate solution. First we will be performing Molish test. Reddish violet ring is present. That indicate carbohydrate is there. So carbohydrate presence is confirmed. Then iodine test is the second step. There are two possible results. Deep blue color polysaccharide starch. Confirmatory tests will be performed like Benedict test. No red precipitate. Then Benedict test, no red precipitate. Benedict test after acid hydrolysis, red precipitate. Then no deep blue color. Maybe mono or disaccharide. Later we will be performing Benedict test. That means red precipitate is obtained. reducing monosaccharide. We know that. Earlier we mentioned the steps. Then red, no red precipitate, that will create non-reducing sucrose. So when we get red precipitate, we will be performing Barfoed's test as mentioned earlier. Two possible results are there: red scum, reducing monosaccharide glucose or fructose; no red scum, reducing disaccharide, maltose or lactose. Then we will be performing this monosaccharide test, that is the Sullivan's test, which will indicate two types of results: either a cherry red color which indicate fructose, or no cherry red color which indicate glucose. Later we will be performing ozone test, long needle shaped crystals for both of this. Okay. Then earlier we mentioned this Barfoed's test indicate no red scum in some cases. That will lead to reducing disaccharides, for example maltose and lactose, which will be confirmed with the help of ozone test. Sunflower shaped crystals will indicate maltose. Powder puff or hedgehog shaped crystals indicate lactose. If we get no red precipitate after Benedict test, it indicate non-reducing sugars. Confirmatory tests, Benedict test after acid hydrolysis, Sullivan's test, etc. are performed. Next, identification of unknown carbohydrate. First set, Molish test. To 1 ml of test solution, 2 to 3 drops of 1-alpha naphthol is added, mixed through the sides of the test tube. Concentrated H2SO4 is added so as to form 2 layers. An appearance of reddish violet ring at the junction of these 2 liquids, presence of carbohydrate is confirmed. First test is positive. An iodine test. To 1 ml of given solution, 2 to 3 drops of iodine is added, no color. That means maybe mono or disaccharide. If there is some color change, blue color, that will indicate polysaccharide. Here no color change is there. So maybe mono or disaccharide. Later we will be performing Benedict test. To 1 ml of solution, 3 ml of Benedict's reagent is added, mixed and boiled for 2 minutes over a small flame. Red precipitate, maybe reducing sugar. Then Barfoed test is performed. Barfoed test is performed in the sense 2 ml of given sample, 2 ml of Barfoed's reagent is added, keep the test tube for 3 minutes in boiling water bath, cool under running water. Phosphomolybidic acid is added drop by drop with mixing until the solution becomes clear. Intense blue color, that indicates monosaccharide. Then Sullivan's test is performed. To 3 ml of Sullivan's reagent, 1 ml of given sample is added, boiled for 30 seconds and cooled rapidly. A red color is obtained, presence of fructose confirmed. The given unknown sample contain fructose. Then next, color reactions of proteins. Proteins are polymers of amino acid linked by peptide bonds. Because of the polypeptide structure and the presence of different amino acid residues, proteins react with a variety of reagents to form colored products. These tests are known as color reactions of proteins. Several of these reactions are of importance in quantitative detection and also in qualitative estimation of proteins and their constituent amino acids. Proteins used in this test are albumin, casein, peptone and gelatin. Egg-albumin is a glycoprotein. Casein is a major protein in the milk. It is a phosphoprotein with phosphopyrrole group attached to hydroxyl group of serine and threonine residues. This egg-albumin and casein are conjugated proteins. So gelatin is formed from the collagen proteins, the connective tissue, by boiling with water. It is deficient in tryptophan and cysteine. Peptones are the partial degradation products of proteins. They are moreover relatively low molecular weighted compounds. Thus, gelatin and peptone are derived proteins. By Urate test. Cupric ion in an alkaline medium forms a violet colored complex with peptide bond nitrogen of proteins. It is a general test for proteins. The minimum requirement for a positive test is the presence of two peptide bonds in the molecule. Individual amino acids and dipeptides do not answer this test. Two bonds is a must. Okay. So addition of excess amount of copper sulfate should be avoided as the blue color may develop instead of violet color. Addition of excess amount of copper sulfate should be avoided. The next, ninhydrin test. Ninhydrin reacts with free alpha amino groups of proteins and free amino acids to give a purple colored complex called Ruberman's purple. I'm not sure about the pronunciation. Ninhydrin is a powerful oxidizing agent which causes oxidative decarboxylation and deamination of alpha amino acids, producing an aldehyde and hydrogenation reduced in ninhydrin. Hydrogenation is that reacts with ammonia and another molecule of ninhydrin to give Ruberman's purple colored complex. Alpha amino acids plus ninhydrin, aldehyde plus CO2 plus NH3 plus hydrodantin plus NH3 plus ninhydrin create Ruberman's purple. This is one of the tests for amino acids. It is positive for all proteins, peptones and peptides. Proteins and hydroproline give yellow color and they are amino acids. It is also used to detect amino acids in chromatography. We often spray this ninhydrin to observe the amino acids while performing a paper chromatography, for instance. Then xanthoproteic test. Aromatic rings in tyrosine and tryptophan undergo nitration on treating with concentrated HNO3 to form nitro derivatives which are yellow in color. In an alkaline medium, yellow color turns orange due to ionization of phenylalanine alone does not give positive result as it does not undergo nitration. Under these conditions, normally this nitration is the basis of yellow staining of the skin on contact with the concentrated nitric acid. Which one? Xanthoproteic test. Then Millon's test, Coley's test. Tyrosine and proteins containing tyrosine react with mercuric sulfate and sodium nitrate in acidic medium to give a red precipitate. This color is due to the formation of nitrate mercuric phenolate. Then sulfur test to cysteine and cysteine. And cysteine or cysteine containing proteins, these containing amino acids are boiled with a strong alkali. Organic sulfur is converted to inorganic sulfide. Addition of lead acetate to solution forms brown or black colored lead sulfate. Then methionine and does not answer the test as the sulfur in methionine is not released by alkali as it is the thioestine linkage. And Molish test for carbohydrates. Proteins containing carbohydrates as prosthetic group. Glycoproteins, we know that. Answer this test. So perform Molish test with albumin solution. Note the formation of a violet ring at the junction of two liquids. Then Newman's test for organic phosphorus amino acids. Newman's test. The organic phosphate is phosphoproteins is converted to inorganic phosphate on boiling with strong sodium hydroxide solution. Then inorganic phosphate react with ammonium molybdate in the presence of nitric acid to form ammonium phosphomolybdate precipitate which is candy yellow in color. Later, precipitation reactions with proteins. Proteins are large molecules with definite size, shape and charge. Depending on the pH of the solution, proteins carry positive or negative charge. Proteins form colloidal solution in which a shell of water molecule surrounds the protein molecule. Polar groups of the protein tend to attract other water molecules around them to produce the shell of hydration. So the solubility of a protein depends on the preparation of and the distortion of polar hydrophobic groups and non-polar hydrophobic group in the molecule. The water molecule interact with the polar group of proteins depending to increase its solubility. The solubility of proteins in the solution will depend mainly on change and hydration, and precipitation of protein can be brought about by any one of the following. So removing their shell by hydration, dehydration is performed. The neutralization of electric charges is another method. Then denaturation, bringing them to isoelectric pH. With the help of all these four processes, like removing their shell of hydration, which is also known as dehydration. After that, we will be performing a neutralization of electric charges. That is another method. Then denaturation, bringing them to isoelectric pH. These are the methods which help in the precipitation of proteins. So precipitation by neutral salts. And neutral salts such as ammonium sulfate or sodium sulfate are added to the protein solution. The shell of hydration is removed and the surface charges are neutralized, causing precipitation of proteins. This is called salting out. What is happening here? So ammonium sulfate or sodium sulfate are added to the protein solution. The shell of hydration is removed and the surface charges are neutralized, causing precipitation of proteins. This is called salting out. The amount of salt required for protein precipitation depends on the molecular weight of the protein. In general, higher the molecular weight of proteins, lesser the salt required for precipitation. Albumin and gelatin are precipitated by full saturation and casein is precipitated by full saturation. So serum globins, being a high molecular weight protein, is precipitated by half saturation with ammonium sulfate. This property can be used for separation of serum albumin from globulins. So peptone is not precipitated by full saturation. And isoelectric precipitation is another method. Proteins have minimum solubility at their isoelectric pH. Proteins are easily precipitated from their solutions by adjusting pH close to their isoelectric pH. This is achieved by addition of an acid or alkali. pH range of BCG is 3.8 to 5.4. When more acid is added, the pH change are flocculent precipitation is happening, then formed by casein at this isoelectric pH dissolves. Linked to the milk, curding of the milk as such example of isoelectric precipitation. Casein formed curd when milk is curdled. This is a reaction because lactic acid produced by the fermentation process lowers the pH in the isoelectric pH by casein, thus precipitating the casein from milk. And precipitation of organic solvents. Organic solvents are dehydrating agents. Addition of organic solvents such as acetone and alcohol decreases dielectric constant of solvent and displaces some water molecules associated with the protein. Removal of the shell of hydration around the protein molecule, this causes precipitation. For the precipitation of proteins by organic solvents, proteins should be in an electrolyte form. This may be achieved by dissolving the proteins in normal saline. Then precipitation of acidic agents, negatively charged ions like picric acid, trichloroacetic acid and sulfosalicylic acid, etc. protein precipitants. They neutralize positively charged group on protein and cause denaturation, resulting in precipitation of proteins. So sulfosalicylic acid is commonly used for screening of proteins in urine and CSF. Which one? Sulfosalicylic acid. Precipitation of proteins by heavy metal ions. And solutions of lead acetate, mercury nitrate and other heavy metal salts are added to protein solutions. The cations neutralize the negatively charged groups in the proteins, causing precipitation as metal protonate complex. The principle is used for treating heavy metal poisoning salts by Salts of iron, copper, lead, zinc, cadmium, and mercury are toxic because they tend to precipitate normal proteins of the gastrointestinal wall. Raw egg white is used as an antidote in mercury poisoning and then followed by an anesthetic to remove mercury ions. Precipitation by heat and acetic acid. Proteins are easily denatured when subject to heat treatment. Denatured proteins are less soluble than the native proteins. Then albumin gets denatured by heating and forms a white coagulum. By adding acetic acid, it brings protein to its isoelectric pH. Thus, the coagulum intensifies. Acetic acid is used added to urine to differentiate between protein and phosphate. If precipitate persists and deepens after the addition of acetic acid, it is due to proteins. If it disappears, it indicates the presence of phosphates.